buenos días, bienvenidos a la sesión general del mes de noviembre. En esta ocasión nos acompaña el doctor Manuel Rivas. El doctor Rivas es un científico de datos biomédicos cuya investigación se centra en el desarrollo de métodos estadísticos y herramientas computacionales para analizar datos genéticos humanos y abordar cuestiones fundamentales en medicina y biología. Su investigación actual se concentra en 4 temas: generar hipótesis terapéuticas efectivas a partir de datos genéticos humanos, desarrollo de tecnologías para sistemas de salud y aprendizaje integrado, inferir en la distribución global de genes de predisposición a enfermedades comunes y raras, y desarrollo de modelos estadísticos de aprendizaje y optimización. El doctor Rivas estudió matemáticas en el Instituto Tecnológico de Massachusetts y posteriormente obtuvo su doctorado en medicina clínica en genética humana por la Universidad de Oxford. Actualmente es profesor en el Departamento de Datos de Ciencias Biomédicas en la Escuela de Medicina de la Universidad de Stanford y también es investigador afiliado al Instituto Broad en el programa en genética médica y de poblaciones. Sus intereses de investigación son genética humana, modelo estadístico bayesiano y optimización, genómica de enfermedades comunes, biobancos de poblaciones, medicina clínica y su traducción de descubrimientos genéticos. Cuenta con más de 30 publicaciones internacionales y ha recibido en varias ocasiones premios y apoyos para la investigación. Bienvenido, doctor.
Bueno, muchas gracias. First, I'd like to thank the organizers for inviting me to speak with you today. So today I want to share with you my vision, or the vision that we've had for some time in the department, as to what impact or what route genomic medicine will take in the 21st century. Data is increasingly becoming the most valuable resource in the world. I think right now, as you can see from this economist piece, they suspect that in the next decade or century, the world's most valuable resource is no longer going to be oil, but rather data. And this is absolutely true in medicine. In this talk, I'll touch on 3 themes that I hope will excite you about the future prospects of integrating genomic data with medical data, along with new data science technologies. So to that end, I'll touch on 3 themes: population medical data, human genetic data, and data science technologies that can be developed with statistical methods. The department that we're in, biomedical data science, tries to bring all these 3 themes together, and we strive to develop methods and tools that will allow this to move forward. So here, what I want to do is also to share with you a few case studies, in addition to some research projects, where the goal is to move us towards personalized medicine, quote unquote, precision medicine, and health. These are catchphrases that have been routinely used, but I hope by the end of this presentation, you'll be convinced that there are new ways and strategies to get to this point. So our main ambition is to understand factors that drive disease risk, and then to understand how each individual is assigned such risk. I'll use type 2 diabetes as the driving example, the motivating example throughout this talk. The main reason is that type 2 diabetes is a disease that affects predominantly in Latin America and predominantly in Mexico in such a way that is going to be a huge epidemic that will affect almost everything we touch in our life. As you can see here, what I've tried to do is to highlight some data points. One is that type 2 diabetes is becoming a worldwide epidemic, or if not, it already is a worldwide epidemic. It affects approximately 300 million people worldwide and is increasing due to changes in lifestyle and diet. Type 2 diabetes is a leading cause of cardiovascular disease, kidney failure, blindness, and premature death. And our current approaches at prevention and treatment is quite slow, and the strategies that we have to date fail to arrest, let alone reverse, type 2 diabetes. The prevalence of type 2 diabetes is projected to grow from over 280 million individuals as of today, to over 400 million individuals by 2030 around the world. As many of you are familiar, a lot of the way we think about human genetics in the context of human diseases is analogous to thinking about a single gene that impacts a disorder, which tracks along the family. So when we talk about human genetics in the context of diseases, we mainly think about Mendelian or rare diseases. Examples of those diseases in the context of diabetes include the lipodystrophies and insulin resistance. Here's an example of lipodystrophy where mutations in AGPAT2 manifests itself in phenotypes including fat loss and varied fat loss. Whereas you have other lipodystrophies where mutations in these genes called lamin A and lamin C exhibit different phenotypes. And those are the types of disorders where we're more familiar with tracking them to a single gene. So another property that we're becoming familiar with, or have already become familiar with here, is that common diseases also run in families. If you have type 2 diabetes, you can ask the question, what is the risk to your neighbor, someone that's unrelated? And that answer is approximately 10%, which tracks with the prevalence of that disease. If you ask the question, if you have type 2 diabetes, what is the risk to your sibling, meaning your brother or your sister? And that answer is approximately 30%. In the context of identical twins, if you have type 2 diabetes, the answer to what is the risk to your identical twin of having type 2 diabetes is over 50%, strongly suggesting that variation in DNA sequences influences type 2 diabetes risk. So one of the opportunities that exists right now is to take information or data from the population and translate that to clinical medicine. And that is the hope thus far. Getting to this point from where we're identifying the genetic drivers of disease risk to identifying therapeutics or providing diagnostics is still one of the outstanding challenges in our field. And I think it's a great time for all of us to be engaged in this activity. The hope is that by successfully mapping the genes of disease, we'd be able to better understand the molecular underpinnings of, or the drivers of disease risk, and thereby being able to design new therapies based on that information, or come up with more advanced ways to identify which individuals in a population are at risk for a disease. So, to make such studies possible, there's a few things that need to happen. And I think now we're at a stage where many of these things have happened, and there's opportunities in the next decade or so to participate in activities that will push this field forward rapidly. So public databases of human genetic variation was essential to being able to map out the genetics of common diseases. Lab and statistical tools to measure genetic variation had to be developed in order to understand what are the drivers of disease. We needed to follow up in activities that will try to identify or discover genetic variants influence disease by applying these approaches. And lastly, you need to set up laboratories in order to understand the biological function of these novel genes and variants that have been implicated. Public databases now contain approximately 99% of the sequence variants in each individual. This has been driven mainly by 3 projects that I'll highlight here. One is the SNP Consortium, which was published right around the time the Human Genome Project was released. The main idea of the SNP Consortium was that we would get an initial understanding as to how humans differ in the genomes from person to person. And by doing so, establishing a catalog that will help us move forward to use that as a technology to map disease genes. The second project that drove a lot of this activity forward was the HapMap project. The idea was at that time, if you had a collection of genetic variants across the genome, could we better utilize or understand the way these genetic variants track with each other so as to efficiently use that information to map disease genes. So these are, quote unquote, what are referred to as haplotype blocks in the human genome. And now, just to go back, one of the main limitations of these activities is that it's largely concentrated on common genetic variants, meaning variants that are commonly shared across the population. The third project that did drive a lot of these activities forward was the 1,000 Genomes Project, where we ended up getting a better understanding as to how, if you could do complete whole genome sequencing in a human individual across thousands of individuals, how that landscape of genetic variation differs from the information that we had. Now, just to go back to type 2 diabetes, these three technologies, or these three databases, public databases that were available, were successfully used over the course of 1.5 decades to map out over 80 genes or regions of the genome that contribute to disease risk. Most of these, as you can see, in 2000, the only one that had been implicated in common types of diabetes was PPR gamma. And by 2014, that collection increased to over 80 or so regions. Now, one of the examples that is really one of my favorites, and it really highlighted the value of Latino populations, and especially more specifically the Mexican population, and I know that many of you probably contributed to this study, and were led this study, was the identification of a novel risk factor for types of diabetes in Latinos, more specifically in the gene SLC16A11. Now that gene was in no one's radar by the time this study was conducted, which really highlighted the value of going into different populations, especially increasing the diversity of populations represented in these genomic studies. Now, just to summarize, each copy of those variants that was associated in SLC16A11 was associated with a 25% increased risk of type 2 diabetes. Those genetic variants are very common in Latin America and extremely rare in other populations around the world. And this study that was published by the Sigma Consortium suggested that about 25% of the health disparity of type 2 diabetes in Latino populations compared to non-Latino populations was explained by this simple region here. Now, the next thing that I do want to highlight is how we can start using human genetics as a tool to guide us in the selection of drug targets. I think one of the things that you're more familiar with is how genetics can guide us to identifying genes or genetic variants that drive disease risk. But now there's a great opportunity to use that same tool to identify drug targets that can move forward in the pharmaceutical pipeline. The strategy would be the same. You'd collect a population along with their medical record data. You identify genetic variants, not that increase risk, but rather genetic variants that lower risk for disease. This gets people excited because then it provides a way to pursue a lead that would allow us to follow up how to mimic that protective allele. Now, there are a few or a handful of examples of protective mutations. The most recognized example is the example of PCSK9 for LDL and myocardial infarction. The story there is that loss of function mutations in PCSK9, if an individual carries one of these mutations that completely knocks out the gene, their LDL levels are extremely low and they are protected from myocardial infarction. Now, one of the interesting properties of these genes was that those mutations were primarily identified in African Americans. If you go outside of African Americans, you'll take a long time in order to identify its association to disease risk. Other examples that have been highlighted include NAV1.7 for pain and CCR532 delta for HIV, which protects individuals from getting HIV infections. Now, the one that has been the most successful to getting from gene discovery all the way to therapeutic pipeline and successfully moving past phase 1, phase 2, and phase 3 clinical trials, at least in the United States, is the example of PCSK9 for LDL and myocardial infarction. Now, rare strong acting alleles of the GWAS regions that we've identified in the past 1.5 decades, what I want to show you guys is that it does provide some interpretation of the GWAS results. One of the challenges with genome-wide association studies is that it's been extremely challenging to identify the genetic variants that drive that disease risk. In addition to that, it's been extremely challenging to identify the genes that implicate the reason why that region is implicated through GWAS. With rare variants, at least with some examples of rare variants, we've identified genetic variants that directly pinpoint to genes that are implicated in the disease. Here's an example of a mutation in the gene called CARD9 that completely disrupted the function of that gene, and it introduced about a 4-fold protective effect against Crohn's disease and ulcerative colitis. Now, this study started in 2008. It was published in 2011, and as of this year, about a month ago, a study was published that showed how a molecule can be identified that mimics the protective effect of that mutation, suggesting that the next step should be in moving this to clinical trials. A second example where we identified a genetic mutation that also protected individuals from a particular disease or common disease was a loss of function mutation in the gene called RNF186. We found the mutation to confer about a 3-fold protective effect against ulcerative colitis. Now, the beautiful thing about this story is that by asking our colleagues and collaborators around the world whether or not anybody had access to genetic data for this mutation, one of our collaborators in Iceland specifically said, actually, this mutation is about 3-fold enriched in Iceland, and we only need about 1,000 cases to replicate your signal that you're identifying. T2D Genetics, which is the company that drives a lot of the genetics research in Iceland, was able to easily replicate the signal that we identified. Now, the beautiful thing is that now we have 2 examples that we can move forward with from human genetics that allow us to then ask the question, can we mimic what these experiments of nature are doing? In the context of type 2 diabetes, there's one example as well, where they've identified this type of signals that you'd want to look for if you're looking for genes or variants that have effects consistent with protection. And that is the story of loss of function mutations in SLC38A that protect against type 2 diabetes. Now, the story here goes that in aggregate, there are 12 loss of function mutations associated with approximately a 65% decreased risk of type 2 diabetes. That story also followed the similar patterns that we had observed in IBD, in inflammatory bowel disease. And that is that many of the primary researchers driving some of the studies went ahead and asked their colleagues and collaborators around the world, does anybody have genetic data for the gene SLC38A that they may want to contribute to move forward the identification of signals that may guide therapeutic development? Now, the next thing I want to showcase is the value that population medical data combined with human genetic data can have in empowering novel data science technologies. Now, population cohorts enables the study of shared genetics with other diseases and phenotypes. One of the first projects out there that allows many researchers around the world to get access to, I think anybody here actually can get access to this data. Anybody anywhere else in the world can get access to this data. As long as you have some affiliation with some institution, getting access to this data from 500,000 individuals where medical record data along with human genetic data is available can be downloaded, analyzed, and queried in order to understand how human genes drive disease risk. This project is the UK Biobank, which has approximately 500,000 participants. As a result of getting access to that data in my lab, for example, one of the things that we've done is to start building tools that would allow other researchers, clinicians, biologists around the world to simply query the association results that are available from that data. We've also added other applications that are featured at the top bar. And in addition to that, we also include video tutorials for anybody that's interested in learning as to how the folks who developed this application use this application for guiding their scientific activities. The Global Biobank Engine was developed by my lab, and the hope is that as we continue developing this tool, other researchers around the world will also engage in its development. The plan is to have this as open source and widely distributed so that anybody who's engaging or moving forward with the development of human genetic activities combined with population medical data can go ahead, download the code, and support their own data sets. As I mentioned, there's also a video tutorial associated with it that you can also find it in YouTube. And there's a 5-minute primer that describes how you would use the Global Biobank Engine. The development of this engine was largely motivated by the development of EXAC or NOMAD, which provides information about the frequency of genetic variants across the world, and the inflammatory bowel disease exomes browser that I developed while I was at the Broad Institute. It includes some applications where you can study the power to detect genetic signals. For example, if you're interested in pursuing a type 2 diabetes study in Mexico for 10,000 cases and 10,000 controls, and you want to estimate the risk effects that you can detect using that study design in the population of Mexico with loss of function mutations, you can go ahead and do that. Here, what I highlight is the example of PCSK9, where we look at myocardial infarction with a disease prevalence of about 10%, heterozygous relative risk of 2, 25,000 cases, 25,000 controls, some type 1 error rate that we'd like to control. And you can see that across the populations that we have genetic frequency summary for. The population where we have best power to detect association is in African Americans, specifically for this gene. Furthermore, we provide estimate of global power to detect association for loss of function mutations across different populations at different parameters, if you'd like to pursue that query as well. In the context of Mexico, you can go ahead and use the AMR row to get a better feel as to what power exists in different study designs. So thus far, the Global Biobank Engine has received very positive reception from the field and the community around the world. We've had over 1,500 users. It was released in August, and over 3,500 sessions, where each section is approximately 8 to 10 minutes. So it means that people are really spending some time browsing the data and getting used to the applications that we're developing. As I mentioned before, population cohorts enables the study of shared genetics with other diseases and phenotypes. The reason being that you have access to medical data, not only about a specific disease, but also about the trajectory of diseases, in addition to other diseases that you may not have directly been interested in. In the context of the UK, we can use that data to estimate shared heritability. is the heritability of type 2 diabetes shared with other traits that or phenotypes that we have access to. For example, type 2 diabetes has strong shared heritability with body mass index, positive correlation. Strong heritability shared with myocardial infarction, hypertension, high cholesterol, and gout. Whereas it has an inverse relationship with the genetics of other phenotypes. For example, it has inverse relationship with the genetics of physical activity and dried fruit intake. At least that was measured in the UK Biobank data. If you're interested in browsing it in the context of the UK population, you can go ahead and visit the website or the link here. Now, just to summarize on the example of the genetics of type 2 diabetes, in 2000, the collection, or what we knew or understood about the genetics of type 2 diabetes, was restricted to the MODY genes and PPR gamma. As of 2017, there are over 200 genetic risk factors identified, and the hope is that we can start using that information to identify pathways and novel therapeutic hypotheses. SLC38 is really one of those examples where it's moved past the genetic discovery to now moving towards the therapeutic hypotheses that's in development. And that study was largely led by researchers at the Broad Institute in collaboration with other folks around the world. Now, a third theme that I do want to touch on here is the concept of how you can use genetic information to think about the risk of an individual for a specific disease. Many have proposed that we should search for subtypes of disease X. The question that, in the context of type 2 diabetes, that many have proposed around the world includes, should we move with the idea of moving with 4 subtypes of type 2 diabetes rather than thinking of type 2 diabetes as one? So the pictorial diagram would be that you've identified 4 pathways that for each individual corresponds to that individual having type 2 diabetes subtype 1, type 2 diabetes subtype 2, type 2 diabetes subtype 3, and type 2 diabetes subtype 4. And the answer, at least as it's emerging from many activities that are ongoing, my personal interest is in asthma. My colleagues or professional collaborators' personal interests include type 2 diabetes, and I have other collaborators and colleagues that are particularly interested in myocardial infarction. And a lot of us are now starting to converge in the idea that thinking of each of these diseases as different subtypes is probably not the right approach. Rather, what may be happening, or is possibly the most parsimonious explanation, is that any individual who has a particular disease is a blend of pathways contributing to that individual's disease risk. So, assume that now you have 4 pathways that contribute to type 2 diabetes risk, disease risk, or any other disease, that each individual then is composed of different mixtures or blends of painting as to how those pathways are contributing to you having that particular disease. Now, I do want to share with you some data, but I also don't want you guys to be fixated on the data that's represented. What I would love for you to get out of these slides is rather that using empirical data, you can get to the point of starting to assign for each individual how these pathways are contributing to that individual's disease risk. And that we can start decomposing based on having access to many GWAS being conducted across the world, across many phenotypes, decomposing that into some hidden structure that puts things into pathways. So I'll focus on the empirical pathways with type 2 diabetes as a motivating example. We have access to a large collection or catalog of association statistics that can be decomposed using high-dimensional statistical methods. In this context, and the first strategy that we're approaching with is to use truncated singular value decomposition. You can see that we focused on about 100 principal components, where each principal component explains a different amount of variance for 3 categories that we've analyzed. For example, there's category 1, where we restricted our analysis of the decomposition of genetic effects on these high-impact loss-of-function protein truncating variants. Category 2, which is looking at mutations that are directly impacting the coding of the gene. And category 3, which many of us have been more familiar with, is the genome-wide look at genetic effects. Here, what you can see is that then when you apply principal components on these association statistics, you can start identifying how each of the phenotypes are split across components. For PC1 and PC2, you can see that PC1 is a major driver of variation for summary statistics for a set of traits. Sorry, it's this one, PC1. For PC2, it's this set of traits here. In fact, for PC1 in this analysis, if you can think as to what trait would drive that major component, that first major component is driven by height. The second major component observed here along the y-axis is driven primarily by body mass index. And here you can see how the decomposition allows us to identify genetic variants that uniquely contribute to principal component 1 versus genetic variants that contribute to principal component 2. Now, just to give you a global view as to how this looks like, each component has a different blend of phenotypes and genes contributing to it, possibly identifying pathways. So in the context of the UK Biobank data, we had access to all the medical record data, in addition to having access to biomarker data and some lifestyle measurements as well. So having access to about 1,000 phenotypes, we can start decomposing how the genome affects components that may be shared. So here you can see that each one of the principal components has a different palette that it starts painting its component with. In addition to that, you can see that each principal component has a different palette of genes that it uses to paint as well. If we focus on type 2 diabetes, there you can see that there's a primary component driving type 2 diabetes risk, but in addition to that, there's a set of other components that contribute to disease risk. Component 1, we identify as the core component. Component 2 has a different blend of phenotypes contributing to it. In addition to type 2 diabetes, you also start seeing gallstones and cholecystitis. And here you see the drivers of that component. Component 3 also has a different mixture of genes and phenotypes driving it. And component 4 as well has a different mixture of genes and phenotypes driving it. And the nice opportunity is that this is a new way of thinking about human genetic data combined with phenotype data. And we're moving forward with different approaches to understanding how the genetics of various phenotypes end up having shared components and how you can think about this in the context of an individual. So just to give you a highlight as to how you can think of this in the context of individual, we have some preliminary data that suggests that you can start painting individuals with these components. For example, these are type 2 diabetes cases in the UK Biobank data that have a score assigned by projecting that individual to these components. And each of these individuals was painted in different ways with those palettes. What we've tried to identify here is, now that we've identified sets of individuals that are at the extremes of these loadings or projections of type 2 diabetes risk, can we identify individuals that don't look like every other type 2 diabetes patient in terms of loadings? And here's a strategy where we've identified individuals here, for example, that although that individual has been painted with a whole bunch of these components, you can see that that individual has very limited contribution of component that's labeled blue or that's colored with blue palette. Whereas with other individuals in the original analysis, you do see almost a uniform sampling of the blue component. In the context of asthma, which I think is a bit clear, just because we have better data or biomarker data in the UK Biobank data at this moment in time, eosinophil count and the platelet crit are major drivers of the brown component. You can see that asthma cases are largely painted with that eosinophil count and platelet crit component. If you focus on the tails of individuals that are separated from every other asthma cases, you can start picking out individuals that where eosinophil count and platelet crit are not the major drivers of disease risk. So here you can see that it makes up a large proportion of the risk to an individual's having asthma, whereas for these individuals, we've picked out individuals that have very limited contribution of the eosinophil count and platelet crit component. So just thinking about how to move forward with some of these activities, there's great opportunities to think about this in the context of collecting population medical data along with human genomic data to move us towards the direction of where we're thinking about a single individual as well. Now, just to briefly summarize some of the activities that we've moved in my lab, thinking about precision medicine, one of the things that we've done is to move forward with a phenome-wide scan for strong effect alleles. What we're interested in is, can we identify other stories like the PCSK9, like the SLC38 example in types of diabetes? So we pursued this study where we focused on these protein truncating variants across 337,000 individuals in the UK Biobank. We combined the genetic variant data from these individuals along with the hospital inpatient record data, the verbal questionnaire data. So the UK Biobank had collected questionnaire data from them, just asking them, is there a history of cancer or type 2 diabetes in your family? In addition to that, just asking them straight out whether or not you have asthma or any of these other diseases from a drop-down menu. In many cases, the verbal questionnaire data was much more informative than any of the data we could get from the hospital and patient record data, which really highlights the value of being able to quickly digitally phenotype individuals at scale. Furthermore, we did have access to data that they provided from Cancer and Death Registry. Now, just to remind you, this is data that anybody in the world can get access to, anybody who's affiliated with a research institution. We focused on about 20,000 of these strong effect genetic variants, and we assessed their medical relevance. Just to give you highlights of the results, we identified about 27 of these strong effect alleles having some association to a medically relevant trait. For those sets of signals, we tried to identify whether or not there was any relevance to other phenotypes that we could get access to. In addition to that, there were some individuals that not only carried one copy of those mutations, but had 2 copies of those mutations, which meant that they didn't have any functioning copy of these genes, alluding to a human knockout. I think many of us are familiar with mouse knockout models. The cool thing, or the exciting opportunity that exists, is that there exist many individuals around the world that, for any particular gene, except the embryonically lethal genes, have their genes completely knocked out. How great would it be to just collect all of them and be able to study as to what their medical impact was? So a few things I did want to highlight is the type of cascade of effects that exist once you move from common variants to rare variants in the population. As you can see, for these predicted strong effect alleles, for common genetic variants, the effect size, which is shown on the y-axis, was quite small, meaning it's close to 0 in terms of the effect size. Whereas when you move to rare variants, you start seeing that cascade phenomenon where, although most of them do contribute almost 0 effect to disease risk, you start identifying alleles that have pretty strong effects. context of muscle or soft tissue injuries, we identified a loss of function mutation in NAL3 that impacted strongly your risk to having those injuries. In the example of eczema and dermatitis, many of you are familiar with the gene FLG, where we identified individuals that carried these loss of function mutations that had a strong effect on disease risk for eczema dermatitis. And here is an example of a protective mutation that we identified in the gene called IL-33 that strongly confers protection against asthma risk. Now, one of the examples that I did want to move forward with in terms of thinking about, well, somebody has gone ahead and identified its association to a particular disease that they were interested in. For example, in IFIH1, a study was published where identified strong protective association of loss of function mutations in IFIH1 against type 1 diabetes. Our interest was in understanding how those genetic variants would impact disease risk for other diseases that we would have access to. In the context of IFIH1, we saw increased risk for ulcerative colitis, so that's shown here, where you see it above, moving towards the right of 0. Protection against psoriasis, so that's shown here, where you see it moving away from 0 towards a negative sign, and decreased risk for hypothyroidism, which is shown here, where you see it moving left of 0. So that's quite nice in being able to ask, if I'm really moving forward with the therapeutic hypotheses of knocking down IFIH1, what would be the downstream consequences that I should expect to see in the context of other diseases? One example where the effects in human knockout individuals, meaning individuals that had 2 copies of these mutations versus 1 or 0, was in the gene called FUT2, where if you did a genetic association study, you would identify its association to many of these diseases, but without going further deeper or diving deeper into understanding as to whether or not it's driven by a particular genotype, meaning whether or not you're homozygous, heterozygous, or homozygous non-alternate, or having that loss of function mutation, is worthwhile pursuing. So here, in this context, what we showed is that the disease risk for this FUT2 human knockout only exists for the FUT2 gene, only existed when an individual was a homozygous knockout. So here you can see that if you only had 1 copy of the mutation, for most diseases, the disease risk was very near 0. And when you looked at individuals that had 2 copies of the mutations, the disease risk was separated from what you'd expect under additivity. So you can go ahead and find this study in bioRxiv, and you're more than welcome to read it and participate in any of the discussions of these studies. But I do want to just highlight the opportunities that exist from combining large-scale medical record data with new strategies of looking at these data sets combined with human genetic data. Now, the last thing I do want to focus on is on building new technologies. So although I do have a strong training, I have a training in mathematics and in human genetics, one of the things that I realized, and speaking to many other folks around the world, is that the main limitation now as the cost of sequencing or generating genetic data rapidly decreases is going to be our ability to get access to phenotyping or medical records. So that motivated us to start building new technologies that would expedite this activity. So one thing that I did want to focus on is on a project in the lab where we're building out tools that we are planning to make open source. Anybody can contribute to the design. Anybody that's interested in thinking about it in the context of medical record data separated from human genetic data or in combination with human genetic data can go ahead and contribute and participate in these research activities. So the motivation for this clinical note tagger is filling out some structured data in EHR is a drain on resources. In the United States alone, clinicians spend a large fraction of the time annotating what diseases an individual has. In fact, most clinicians report that over 30% of their time is spent on sitting down at a computer and writing down disease codes for any given patient. And I think that the estimate nowadays is that out of the 8 minutes that the clinician has with the patient, 4 of those minutes, and that's being kind, the physician spends on a computer rather than with the patient. And this is the case as well in the UK and other parts of the world where digitalization of medical records is being introduced. So our main motivation was to think about, well, what opportunities in new technologies that are being put forward can we propose that would decrease the amount of time that a physician spends with a computer and increases the amount of time that a physician spends with their client, or sorry, with their patient. So the goal of the clinical note tagger is to assign ICD-9 codes to patients using their clinical notes, reducing cognitive workload, and standardizing bill practices in the process. In other countries, of course, this is separated out from an insurance-based procedure, but rather more of a centralized national public health program. In the context of the UK, in the National Health Services, the ICD codes are not used for billing purposes, whereas in the United States, they are used for billing purposes. So the methods that we've started to introduce is using this clinical note tagger developed in a deep learning algorithm where you take unstructured text from either voice as input, scanned notes, or typed notes. We train a machine learning model to then identify ICD codes and project them on into new clinical notes that are available with unstructured text. These results yielded, from our initial pilot projects, the results yielded some accurate prediction of ICD code categories, and we're in the process of moving forward with how we would bring in voice technologies to replace the property of a physician having to spend time, 4 out of his 8 minutes on a computer, and rather just dictate the interaction or the diagnosis with the patient. So ALTUD is the software that we're developing. The plan is to have it open source and to make it available to anybody around the world. And the proposal is to develop this platform for the analysis of text-speech for understanding disease, where we not only bring in medical record data, but we also bring in human genetic data and new modalities for bringing in that data and integrating it together. So in conclusion, one of the things that I do want to share with the audience is there's ample opportunities in linking medical population data with human genetic data and new data science technologies. The opportunities that exist is in generating effective therapeutic hypotheses so that we move forward with properly introducing some of our new ways of thinking about human genetic data to design effective therapies. And this is what the catchphrase of precision medicine has been all about. But few examples exist thus far that have gotten to the stage of moving past functional understanding of characterization of those mutations. And the truth is that by ample sharing of data and ample collaboration, we will expedite some of those activities. The second is opportunity of assigning individual risk to disease, which many folks use the catchphrase of precision health. If you understand that certain fraction of the population is at increased risk for type 2 diabetes, there may be other interventions, low-cost interventions that may be introduced. But without that information about what risk exists to disease, it's quite difficult to get to a point of proposing interventions. And the third strategy that I've alluded to that is really new methodologies and new ways of thinking about the human genetic data combined with the phenotype data is identifying pathways contributing to an individual's disease. So this is more analogous to or matching to the catchphrase of personalized medicine. Assume that you have asthma patients where In fact, eosinophil counts have nothing to do with its predisposition to disease risk. Then, prescribing drugs that target eosinophil counts may not be the right approach. So that's the opportunities that exist, and to finish off, I think there's a great opportunity for Mexico to be one of the leading partners in our global search for understanding, treating, and preventing disease. With that said, I'd like to finish off by thanking the researchers and members of my lab. In addition to that, also acknowledging the contribution of many of my mentors that have led most of the research, some of the research studies that I alluded to here in this presentation, including David Alschler from the Broad Institute, Mark McCarthy at Oxford University, Mark Daly at the Broad Institute, and Carlos Bustamante, who's the chair of my department. That said, thank you.